Hello there, I'm Stephania. I studied how the reproductive cycle of deer mice impacted their skin bacteria. Now, I'm sure most of you are wondering why. Well, I was inspired after reading some articles of bacteria attraction. The articles concluded that skin bacteria activity, such as odor production and quorum sensing, play a role in bacteria attraction. However, I noticed a gap in all the articles. Not a single one used human females for fear that the reproductive cycle would skew the natural flora. Considering that the females consist of half our population, the skin microbiome plays a role in meal selection for vectors, and vectors transmit diseases like malaria and yellow fever. I believe this gap merits attention. However, working with human females is not a feasible undergraduate research project. There are too many variables to control. In addition, I would need access to the females every day of their cycle for multiple cycles. Dr. Hawkinson recommended we study deer mice instead. Deer mice are easily accessible and controllable at UNC because we have a colony at the Animal Research Facility. In addition, they have very short reproductive cycles. They last about 4 to 6 days. In 1 week, we practice animal handling and we practice phase identification. We refined these methods. The next week, we acquired vaginal swabs and pipettes as the mice were temporarily anesthetized. The swabs gave us samples of the natural flora, which we then placed in a nutrient broth and incubated for 24 hours. The pipettes gave us a sample of the cells in the area, which are needed to determine the stage of estrus, aka the reproductive cycle. Throughout data collection, we also gram stained, which helps depict physical characteristics of the bacteria like shape, growth pattern, and possession of an outer membrane. And we streak plated as well, which depicts colony growth and helps determine population size. To conduct the experiment, a list of tools can be found in our research proposal and eventually in our manuscript. To help you better understand the procedure, I photographed the images in the material section. Obviously, we needed mice, a microscope, and an incubator. On the far left, you see the setup for cultivation. In the middle, you see the setup for gram staining. And on the far right, you see the setup for streak plating. The end result thus far is the following chart. There, I inputted the highest, lowest, and mean of the number of forms of bacteria seen in each phase. Most interesting, almost all the peaks are around or at mid-estrus. The mean line tells the most exciting story. It shows 2 peaks, one in proestrus and another in mid-estrus. Whether any of this is significant is yet to be determined, though I find it interesting that all the phases with the most diverse flora are the ones with the most diverse cell types. Below the chart, you will find images of cells on the top and bacteria on the bottom. We hope the pictures help the viewer get an idea of diversity witnessed in these samples. The study had some huge limitations. First, the recent pandemic caused the closure of the university, which prevented us from analyzing the streak plates, which was essential for determining population size. Second, it would have cost several thousands of dollars, if not tens of thousands of dollars, to accurately determine the species of bacteria via DNA, which was never a realistic option for the study. Due to that, diversity can only be determined by distinguishing the physical features of the microbes. Lastly, as of yet, the significance of the data has not been determined, so the results are just speculations at this point. On a positive note, quite a bit was gained from the project. First, we confirmed the estrus phase can be determined in deer mice, which had not previously been done. Methods for obtaining cells and microbes in deer mice is more refined. Some bacteria can be identified by their physical properties, so we gained a better idea of what is naturally found on the rodents. Lastly, if our speculations are true, the study may demonstrate that there is fluctuation in diversity throughout the cycle. For future studies, I recommend conducting the same study with the addition of DNA analysis and calculation of population size. Furthermore, one can also expand it by seeing how vectors like mosquitoes and ticks react to the changes in the estrus cycle in the mice. With that, I would like to thank Dr. Pat Burns for offering his assistance in determining the estrus phases and for providing literature to understand estrus phases in deer mice. I would like to thank all of the McNair staff members for supporting us in undergraduate research. I would like to thank all the research facility staff members for taking care of all the animals. And I would like to thank all the deer mice participants, especially OGDM1. Right here, you can find my references. And now I am accepting questions. Thank you for your time.